Isto está enchufado, ¿sí?
Sí.
Jo pot unir el partit que ho faré fala cap o baix de la història us sona.
Okay. Good afternoon. We are going to start the seminar. So please introduce yourself and let's start. This afternoon we have first the seminar and then we have two hands-on sessions on two of the topics of the school. And the hands-on session are going to be in the computer room that you know that is on the building. And now we start with the seminar, which is going to be given by Tomàs Sintes, who is a researcher here at IFISC, and he's going to talk about growth on some algae in the some plants in the ocean. So so let's start with the talk of Tomàs.
Okay, thank you, Peter. So welcome. The title of the talk, as you can see here, is about non-equilibrium growth processes applied precisely to marine ecology problems. The first thing I probably know that not only scientists but almost everybody has been fascinated along history about the different kind of shapes that can be produced in biological, chemical or physical processes. Just to mention a few of them, you can observe here what's the typical shape that the bacteria colony that is growing in a petri dish forms. This kind of shape is not that far away from the viscous fingering produced in typical heliotrope experiments. In this kind of experiments you have two plates very close together, that is one liquid in between, and you inject another liquid that that is immiscible with the first one and with a different viscosity. And so this is the kind of structures that they form here. Other types of structures that you can observe is this during a storm. This is a lightning or a spark that is produced in an electric medium. Also, a very branched and ramified structure can be observed in something that is called electrodeposition processes, in which if you enlarge or you make a zoom of one of these tips, you can observe a very rich shape as the one is printed here. And this structure is not very far away from the kind of shapes you can observe in geological processes, for instance, in the formation of a glacier. Not to mention the different types of ice crystals that you can find in snowflakes. Or this kind of ramified structures or dendritic growth that can be observed in porous media, in sedimentary rocks. This is just deposits of manganese oxide in these types of systems. Or maybe you don't know what this looks like, but this is just the structure of a city that is growing. And nowadays, people that study this kind of processes is known not as complex city, but as complex city, right? Yeah, complex city. That's fashionable. Who? What? Okay. Okay, fine. Not smart city, complex city.
Okay.
And yes, this guy here is, I found a very nice sentence of this guy here, is Wilson Bentley. That was a farmer that was living in the States in the late 19th century. So even he was a farmer, he was very much enthusiastic about the photography that was just in the very beginnings. And he was able to make up to more than 5,000 pictures of different crystal ices. So he wrote a very nice sentence here that you can read it if you want. Oh, it will be on the web page, so you don't read it now, you can read it later. Now, let's take a look at a nice movie that was taken just from the web, but you can observe here if you look at, this is a bacteria, Streptococcus pneumoniae, how it grows in a period of 12 hours, right? There is some sound there, and it looks like an alien movie, something like that. But so you see, this is the type of growing shape of this bacteria, right? So one of the things that people wanted to understand is how these structures are formed and they grow in time. So, and this problem attracted very much a mathematician that in 1961, he was called Murray Eden, presented a numerical model trying to mimic and understand how these cells grow and reproduce and form these different kind of shapes. So what I will present here is something that I said paradigmatic or characteristic non-equilibrium growth models. And the onset of this kind of models, we can put it in the work carried out by this mathematician. So in 1961, in a symposium of mathematics carried out at the University of Berkeley, he presented this type of model that I will describe in the following, that it's the simplest model you can do. So assume that you have a grid, a square lattice, okay, you make a grid out of it, and you put in the center just what it will be our starting cell. This cell will start to divide, and this will form a colony. How it grows? The growing rules are very simple. These yellow squares are known as the growing sites. All the nearest neighbors squares to the cell, existing cell, are the growing sites. So what he did is just to select at random, with the same probability, which of these growing sites will be occupied by a new cell. And then you have this type of structure, and then you have a new set of growing sites, and you repeat this process ad infinitum. The type of structure that is formed is very close to the movie I showed you before. And what was very much interesting is try to characterize the geometrical properties and the dynamics of this growing structure. If you go back into the literature, starting from the first work of Eden in 1961, you can jump to 1972, where this, with some modifications of this model, were used or introduced by Williams and Piertnes to study the growth of a melanoma, of a skin cancer. This paper was published in Nature that year. If you do another jump in time, these two guys, Pliske and Rast, were able to characterize what is known as the active zone. The active zone is basically what is the structure of the available growing places. How it grows with time, how many of them that you can find there, what is their geometrical properties, etc. In 1987, the width of this active zone was accurately, with new computers, they were able to perform larger simulations, and they observed that the growth of this, or the width of this active site, could be related to the overall size or radius of the growing structure, depending with a given exponent. This exponent is the growing exponent, and was found to be something like 0.37, something like that. This result was compared immediately with a model that was published 1 year before, that was known as KPZ model. It stands for the Carter-Parisian-Zank equation. That was precisely developed to study the growth of surfaces. And just to compare, the one-dimensional model of this KPZ gives a growing exponent that is 1/3, and it's very close to the exponent obtained in these Eden models. So just the basic equation for this KPZ is this one. This is a very famous equation in which is a non-linear, contains a square of the gradient, and this is a diffusive term, and you have some noise. And typically people want to characterize this quantity that is precisely the width of the active zone. And 1 year before the KPZ model was published, Farrington family and Tomàs obtained a scaling relationship for the growth, for the size for the width of these active sites in growing surfaces. So it was able to put, in terms of these scaling relations, and they derive 3 types of exponent. One is called the roughness, the growth, and the dynamic exponent. And precisely the growth exponent is the one that is close to match the one obtained in the Eden model. If you go ahead in the literature and you go to the development of the use of renormalization group techniques, they were able to, let's say, they propose using this kind of techniques that the KPZ model could be the field equation that was able to mimic or reproduce the growth, the development of many types of growing models, particularly the Eden model, but also other types of models that the ones that are known of the growing of surfaces due to ballistic deposition, or something that is also known as surface-on-surface growth. And in fact, only in this latter case, this surface-on-surface growth was proved in a rigorous way that can be represented by this KPZ equation. In this, even in the basic Eden model, we don't have yet a proof that the KPZ equation is able to reproduce this one. So this is the story of this type of models. But there are other things, other interesting things. For instance, 20 years after Eden presented his model, these two persons, Tomàs and Sander, in 1991 presented something that is called DLA. This means diffusion limited aggregation process. What's the main difference between the first one? The main difference is that here we have our seed, and we select at random one of these nearest neighbor positions just to let it grow. In this case, we have the seed at the center, but we now just put second particle in the system and let it diffuse. They perform just a random walk. And then you start from a faraway position from the seed, and then this performs a random motion until it reaches one of the nearest neighbor's positions of its seed. When this happens, you will see that the type of structure that is formed, you will not find a compact structure anymore, but you find a quite ramified structure that looks like a self-similar object, or even what is mathematically called a pseudo-fractal. The geometrical properties of the two growing models are in principle completely different. Another interesting result appeared in 1984 in a paper published by Niemeyer and co-authors, in which what they did is they want to study precisely the structure and formation of these lightnings in a dielectric medium. So the difference between this model and the Eden model, the only difference is just instead of selecting one of the growing sites randomly, they were selecting according to probability that is proportional to the strength of the electric field. To obtain the electric field, you need to know at the beginning which is the value of the electric potential in all the space that surrounds the spark that is, or the lining that is forming. Therefore, once you have a complete map of what is the electric potential, you can extract the value of the electric field in the nearest neighbor positions of the spark that is forming. So now you grow in a direction, in whatever direction, but with a weight, with a probability that depends on the strength of the electric field. When you do that, the kind of structure that you form is very much similar to the one developed in this kind of DLA models. Therefore, it is established that when the growth probability depends on the global structure of the cluster, and it's not anymore a local property, we can say that if you have a scalar field, in this case the electric potential, that obeys the Laplace equation, this can be simulated as if it was a random wall, and he uses the same boundary conditions. In a dielectric media, the electric potential fulfills the Laplace equation, and in the case of a random wall, you write the probability that a site is occupied. It's just a discretized form of the Laplace equation as well. So all these kind of growing processes are known typically in literature as Laplacian growth models, because there is a scalar field that fulfills the Laplace equation. We can go still further in the literature, just to finish. If you, instead of having a fixed seed that is growing, you have several of them that all of them are diffusing, what you will observe is that you have this kind of objects that they move all together, they diffuse, when they interact, they are able to become tied together to chemically react, whatever you want, and they form large clusters that are diffusing and colliding, and they coagulate and form larger and larger structures. This model was able to reproduce, for instance, what is the formation of soot particles during the combustion in a flame, right? These are soot particles that can be obtained in a flame. This is just a single one of them. And from the geometrical point of view, they are comparable to this kind of processes. Just to study, for instance, the probability distribution of finding a cluster of a given size, the time evolution of this quantity, this is the number or the concentration of clusters containing K particles, the time derivative of this one, it can be derived from an equation that was proposed from this guy, von Smoluchowski, in 1916. Essentially, the physics is contained into these kernels. This is called the coagulation kernel. And in principle, you have a set of an infinite first-order differential equations that are coupled all together. So this is a problem that in very few cases you are able to find an analytical solution. Just only when these coagulation kernels have a very simple mathematical aspect. And we can even complicate this expression a little bit if we assume that at some point these clusters can break. And so we have to introduce something that is related to the fragmentation processes as well that make things even more and more complex. This is just a presentation of a very, let's say, important hits in history in this last, let's say, this half century on these growing models. And if you are interested in this type of problems, I suggest you to have a look at this book that was published by Paul Mectin in 1998. And you can even find one of the last versions was edited and updated and edited in 2011. It's a large book, close to 1,000 pages, where there is a summary of almost all what people have done with this kind of models. So there is a lot done. But I was going to talk not about the growth model, just a sketch, just to show different things that people can be done and how things can be analyzed, but also to try to apply this kind of techniques to the growth of clonal plants or to marine ecology. The first thing I have to say is, what do we understand by a clonal plant? So in principle, what we have is plants. If they are clonal, we can think that they are beings that share the same genetic code, right? And this is exactly what it is. In fact, we have a single plant. We have a single plant, and in particular, this picture is very nice. And we have a root here. This is called the rhizome, and this is underneath. That's below, right? And what you see on the surface is just these branches. Of course, all of them belong to the same plant, and all of them share the same genetic information. These guys here, this one, this one, this one, this one, these are called clones. Okay? We are able to find in our seas a large number of species. In particular, these are seagrasses. Okay? We can find a large number of species. In the Mediterranean Sea, the two major ones are called the Posidonia oceanica, and this one is Thymolothea nodosa. Well, and as I said here, they are very important from the ecological point of view because of many things. For instance, just talking about Posidonia oceanica is able to cover about 2% of the Mediterranean Sea along the sea coast, which means 5 million of square kilometers. It's able to increase the production of biomass, oxygen, contains a lot of small animals. It's called epifauna, small snails, etc. Is the natural habitat for many types of animals. And the presence of this network, these roots that form complex structures below, is able to prevent from erosion, create natural barriers that is able to protect our beaches, etc., etc. So from the ecological point of view, it's a very interesting system. But not only seagrasses are clonal plants. If you just go into your laptop and type clonal plants, you will find thousands of species, not only in the sea, but also terrestrial ones. So they are just a few set of, most of them you have probably seen. This is geranium, this is spargus, this is minta, salvia, these are juncus, right? These are giants. This looks like trees, but these are giant mangroves. Okay? And this is also an alga. As Pera said at the beginning, these seagrasses are not alga. We are really plants. Okay? But this is an alga. This is also a clonal plant. So you can find thousands of species that behave like that. If we want to characterize the growth of these kind of structures, let's say we have to create a model to mimic the growth of these clonal plants. So I took one of the previous pictures I showed you in the last slide, and despite biological systems are very complex and there are a lot of parameters to model the growth, it's important to realize that a few of them control the growth of these plants. For instance, we must know which is the velocity at which the rhizome grows. This is called the rhizome elongation rate. Also, an interesting quantity is that different shoots are separated on the rhizome at more or less a rather regular distance. This is called the spacer length. Other two important facts is that they are able to develop lateral branches, and this branching is characterized by a branching rate with a characteristic branching angle. And finally, each of these shoots might die with a given mortality rate. Okay? These parameters here are characteristic of each of the species I have represented. To any of the species have characteristic values of these quantities. So, taking these parameters as our simulating basis, let's try to do a model that looks like an Eden model but applied to this kind of clonal plants. So, we can start with the first shoot with the rhizome pointed in the direction of this arrow here. In the next step, with the distance that is related to this spacer length, we can have a second shoot that continues growing in this direction, and eventually it might branch with this branching frequency and with the characteristic branching angle. In the next one, it could happen that now we select this green arrow here and we will grow in this direction, can continue growing in this direction, and eventually may branch in, for instance, this direction, right? So we can continue, we can iterate this process as much as we want. And also a special issue is just we have to take into account a kind of exclusion area around one of these shoots, just to take into consideration that these shoots must compete to get nutrients and light, so they cannot be as close as possible. A way to estimate this exclusion area, we can get it just because in a seagrass meadow we can obtain a value of the density of shoots, the number of density per square meters. So if we know this quantity, we can adjust the value of this exclusion area. We can easily estimate the value of this exclusion area here. So if we do that, for instance, if we put it into a numerical model, what we can observe is that this will be how this plant grows and extends into the sea. So since we don't have much time, and when we have that, we can do a lot of things in order to characterize this kind of structure. For instance, you can see here, this is the shoot density, the number of units per square meters that we have. You see that, and this is in years, so this is simulated for 15 years of growth. You see that at the beginning, the first 2-3 years, we find the structures that have very low density. Suddenly, they grow very fast, and we have structures that they reaches a plateau. This is the density that these plants are assumed to have when they are old enough, right? So, and we don't have a continuous or a smooth transition, but we have a rather sharp transition from very low density structures to very high density structures. In this region here, where we have very small number of units and there is no essentially spatial dependence, the number of units grow exponentially with time. But finally, if we have very dense structures here, then the spatial dependence is crucial, and they just grow, the size of the growing cluster just grows. Therefore, the number of shoots, it just grows with with the power of 2 of the radius of the structure. So you can see different snapshots of how this structure will be at different years. We are also able to characterize which is the characteristic time at which this structure will jump from low to high density values that is obtained with this expression here that contains one of the 2 parameters. Okay, this is just the characteristic branching angle, and this is the branching frequency here. We can even do more things. For instance, we can measure the amount of biomass produced. We can measure the number of units with time. This is the region in which we observe this exponential growth. And we can also analyze the growth rate, but the growth rate of the whole patch, of the whole system, right? The bars here and crosses, this corresponds to experimental observations of one of the particular species that we have simulated, and you can see that reasonably fit with our simulations. I can show you data, for instance, in this case, from another species, this is from Posidonia oceanica. You can see also the low and high density region and the number of units. This is the region where we observe an exponential growth, and this is the quadratic growth in this red line there. Okay, but what else? So what else can we do with this type of models? And for instance, we can do several things. This type of models have been used in restoration programs. We have used this kind of models also to study how relevant they are in order to sequestrate carbon or carbon dioxide, and how important would be the impact of the presence of this ecosystem in the climate change. We can study the genetic diversity, and this is a measure of the level of adaptability of this kind of species to environmental changes, and also to study the formation of patterns in very large structures. So just to mention a few of them, so we use this kind of program in a European project in which we were studying, well, it was a restoration program in the natural park here close to Lisbon, here in the Arrábida Spichel. So we were simulating, we want to recover the ecosystem that was present in this area here, which type of endemic species we might use in order to succeed, in order to restore the natural environment there. So this type of models are able, so we use different kind of species, they have different set of parameters, and we use different type of planting strategies. So, and using this kind of problem, we are able to see that if we use, for instance, this type of species here, we can see that starting from a planting unit, it's very hard to see from your place probably, but with a small one, it can very quickly after, this should be 0.4, this is a few months, 6 years or 20 years, how it will grow. So we can look at the number of shoots, we can look at the density, etc. So in principle, we might think that this could be a good candidate to restore the ecosystem at that place. We tried other endemic plants. This is too bad. So for instance, in this case, you can see that there is almost an exponential decay in all the quantities here. In this case, this kind of plant will not do it. So we can help the marine ecologists there to suggest which plant will do it better. Other things about the sequestration of dioxide, carbon, or carbon, we are also able to measure the amount of carbon they are able to sequestrate in a certain number of years. So the key point is that more or less well established the amount of grams of CO2 that these kind of plants or these kind of ecosystems are able to retain per square meters and per year. So if we know how fast these plants are growing, how large is the extension that these plants occupy, we can make estimates of the amount of carbon they are able to retain or sequester, right? So if you do it, just to have an idea about the numbers, if we look for a fast-growing species like this Haloludi wrighty that grows in Australia, you can see here that if you start, for instance, we have to put some boundary conditions, of course. If you start, for instance, with a plot which has a size of about 1 hectare, with a number of units there, you can say put a few hundreds of units in 1 hectare and let it grow. You will see that if you let it grow after 50 years, it will be able to sequestrate up to 3,000 tons of carbon dioxide, just starting with a plot of 1 hectare. You can do it in several of them. So, and this is a quite relevant quantity. We can do something more that, for instance, analyze the genetic diversity, and as I said, and this is a measure of the adaptability of these systems to environmental changes. So in principle, that I said that these plants are clonal, and which means that they have the same, all the shoots have the same genetic information. However, they have sexual reproduction. So they have flowers, seeds, they can reproduce sexually, but also there are mutations that take place during this growing process. So it is known that approximately the meristematic cells that divide at the tips of the rhizome, we can observe about 1 mutation over 1 million reproductions. So every 1 million reproduction, we are able to take something that has some genetic differences. Right? So we wanted to study also this genetic diversity. And why we wanted to do that with this type of models? For a very simple reason. This is experimental data or observational data. The first thing that you must focus in is the number of units that people sample in this kind of experiments. You see, this is for different type of sites and different type of species. You see that the number of units that have been sampled just go up to 40, 50, just a few dozens of them. In the best cases, they are able to sample a few hundreds. Of course, if they sample thousands of them, they just kill it. So they cannot sample a large number of them. Good. The second column that is quite interesting is the different number of genotypes they found. They found, if you compare with the number of units that have been sampled, they found a large number of different genotypes. Therefore, a measure of what is called the clonal diversity, which is just the ratio between the distinct genes with respect to the number of sampled shoots, it's a very large number. So the point is, from the statistical point of view, how reliable can we believe these numbers here? So with this type of models, we can estimate how relevant these quantities are. So if we do that, for instance, with the same species as I presented before, different colors means different genotypes. So you can see that, for instance, in Posidonia oceanica, if you simulate the growth for 150 years, you can see that the ratio between the number of genotypes and nodes is much, much, much smaller than the one that they are able to measure when you just take a few of them. In the case of Thostera marina, with structures that are very similar, this is an area covered about 10 square meters. This is very much similar to this one here. This is very similar to this one here. This will give, we expected to obtain a clonal diversity of 20%. If we do that in our model, we observe that it's not 20%, but it's about 0.3%. In fact, this is much, much smaller. So the problem that arises here is the role of the size of the sampling in order to obtain this number. So, and what usually happens is that as you increase the number of samples, the number of genotypes you observe also increases, but the clonal diversity decreases. in this picture, for instance, if this is just a density map of the different type of genotypes you can observe, depending on how many units we sample. In this picture here, we have taken 1 unit in boxes of a size of 20 by 20, okay? This is the type of picture we have. But now, if we increase the sampling box, so we take 4 times less units to sample, this will be the density map. And if we enlarge it to 80 by 80 centimeters, what we obtain is this one. So it's a completely different map, right? What else we can do? For instance, we can look at how the population of genotypes, how it changes depending on how big is the size of the sample. So if you are able to see this arrow here, you can see that in this direction we are increasing the sampling size. This is, we have very few samples and we increase. We have different curves here. Okay, and the latter one, the last one, this one means a complete sample of the whole system. So the most interesting part is that if instead of plotting the number of units, we plot the probability of finding one particular genotypes, we can see that all of them more or less fall on the same line. We have a characteristic slope here. And another thing is that this is just increasing the sampling size. But now if we sample everything, but at different time steps, so at young plant, older, and older, for instance, taking data after 100 years of growth to 100 up to 500 years, and you plot this probability, you will observe that we observe again that all the curves would like to fit. In fact, we find 2 different slopes. One for the most abundant genotypes and the other one for the rest. So in principle, one even could think that we can try to fit this probability distribution of sizes to some, let's say, probability distribution in which we have a small number of genotypes that are dominant and we have a large number of those that are just there with very small population. For instance, we are trying to fit that with a Pareto distribution that usually have this kind of properties. But it's not that simple, right? We can do much more. Other kind of different ways to approach to the richness or the different number of genotypes in the population, and how to extract useful information, and how this information depends on the sample size. This can be done through the evaluation of what is called diversity indices, or also known in the literature as Hill numbers. So, if you look at this at the value of this quantity, the Q diversity index for different values of Q, depending on the number of units that we have sampled, that you can see that for small numbers of Q, the result strongly depends on the sample size. Particular Q equal 1 is known as the Shannon entropy, that maybe sounds to you. And for larger Q values, we see that the result is not really sensitive to the sampling size. So probably we can get very much easy information if we look at Q diversity indices larger than 1, with a small number of sampled units. We're still working on that. And the last problem that I would like to talk about is the formation of patterns in large seagrass meadows. This is a picture taken from Cabrera, an island in the southeast of Mallorca that you probably know. So you can see all this area here. This is Posidonia oceanica. If you look at that here, you can see these kind of circular spots, like the one that I showed you in the movie. Just to make you an idea, for Posidonia oceanica, this structure has about diameter of about 10 meters and needs about 100 years to be formed. So this gives you an idea of how fragile this ecosystem is, and how easy it is to destruct something that needed a large number of years to grow. But also interestingly, we observe not only this kind of structure, but also this kind of circles here. That in principle and for many years, since the island was occupied by, was used by the army doing, shooting guns, right? Yes, okay, shooting big cannons, right? So we thought, or people thought from the very beginning that this was the trace of these shoots, right? This was just the, this was just the target, right? So whatever was there. They are not there anymore, but but these structures are still there. So this is quite interesting, right? This picture was taken several years ago, maybe 10 or 15 years ago, right? But also was very quite amazing that this summer in the newspapers, this was a picture that appeared on the newspapers. This picture was taken in the coast of Croatia, in an island called Dugi Otok, okay, in the Adriatic Sea. That was published in May this year, just before summer, right? Where it's not very easy to see, but you can see here, here, they could find up to 25 circles of about 50 meters diameter. They are quite large. And in relevant and well-known newspapers, it was probably assumed that they were created by aliens. In fact, at that date, people living in this island saw very strange lights in the sky. So that is the most plausible explanation, right? Yes. Okay. But do we only see this kind?
It's a picture from the future, right?
It's a picture from the future.
From 2001.
Oh, sorry. It's 2014. It's my fault.
Some people think that they are fairies, right? Because they're fairy circles.
Fairy circles, yes. But this kind of circles also appear not in the sea, but also in terrestrial ones. No? This is a picture taken from the Namib Desert, where you can see this kind of, here is shown more nicely. You can see this grass. You can see this kind of circles that in which nothing grows there. And in fact, if you have a tree here, you have an area even much larger where nothing grows there. One of our former students, Ricardo Martinez, right? So he did a PhD here, just studying this. Well, studying part of his PhD was done studying these kind of structures. The work was under the supervision of one of my colleagues, Cristobal Lopez, and they were able to explain the formation of these circles here, and why around the tree we do not find anything. So this can be explained with mathematical models, right? So we need for that to have some kind of local and non-local dependencies. And just to finish with that, the way in which we are trying to model the formation of these amazing circles of Posidonia oceanica in Croatia or in Cabrera, in Mallorca, is just using the very a kind of model based on the Ngumo equation, right? In this kind of this equation was derived, if you know about it or not, about the starting from the Hodgkin-Huxley equation for excitable neurons. So this was IFISC was able to rewrite this equation in a more easy way, and it has been used in the literature very much in order to study excitable systems, for instance, in semiconductor lasers or in many different physical systems. What do we need? So, why do we think we can use this kind of approach to study this type of structures? So in principle, this history is quite simple. We have that the time derivative of the density of shoots of whatever seagrass species we have is something that in principle depend mainly with two parameters. One is the frequency of horizontal branching, and this is the mortality rate. So, and at the beginning, we have this kind of exponential growth in the number of units. This comes if we solve this differential equation that is very simple, and if we assume that these parameters have a definite and constant values. But the reality is not like that. And it's not like that not only because these values have large fluctuations and have a strong seasonal dependence, but also because these values can also, these parameters can also depend on the local density. For instance, it's easy to understand that this growing rhizome that is in an area that has a very low density of units, it's much more exposed to erosion, for instance, to waves, to fishes that eat for whatever reason. So they are more likely or have higher probability to die, right? On the other side, if these shoots are placed in a region where the density is very, very high, they have to compete strongly for light and for nutrients. So again, growing in a region that is very heavily populated will be also penalized. And we find a particular region in between of densities not very large, not very small, in which they are optimal to grow. So we can mimic this kind of behavior just introducing a dependence on this, for instance, in the frequency of branching that is given by this term here that has the shape of an inverted parabola. So we have small values for large and small and high density values, and we have high values for intermediate values of the density. We need something else. This will be a local interaction term. We need something else, which is this term, which contributes to a non-local interacting terms. So if we want to reproduce circles of several meters of diameters, we need something else, not just the local density. And what could be the biological explanation to introduce this kind of non-local interaction? This we are not pretty sure of that, and the biologists with whom we are working, it's not also very clear for them, but we have some yes, we know that this kind of plants they have something that is called apical dominance. Apical dominance, this means that you have the rhizome, this rhizome is growing. The tip of the rhizome is where these meristematic cells are growing, okay? And they send some chemical signals backwards to the rhizome. So these chemical signals means that they can prevent this horizontal branching up to a certain distance. And this has been estimated and measured for different kinds of species, not only aquatic ones, but also terrestrial ones. That could be even of the size of meters. So probably this apical dominant effect has something to do with these non-local interacting terms, and could be an explanation of this phenomenon. So if we are lucky to do that, then we are able to say something on the news, probably. And finally, it's almost 4, so just people with whom I've been, or I continue, or I already or still collaborating with them, people from here and people from other parts. So thanks. Hopefully you have enjoyed the talk, and if you have questions, I'm open to that.
Thank you very much.
Do we know if these scopes are still growing? Or
Sorry?
The scopes, the circles are still growing?
No. We don't have such, well, in principle, we don't have this information because it takes a long time. So what we have seen is that they are just there. The size does not change. This is very large. These rhizomes grow a few centimeters per year, so it's very hard to observe them. Yes?
Okay, we have in Baltic Sea, we have this every summer there is this cyanobacteria problem. There's a lot of blue algae there. Do you know if there have been any kind of modeling on that?
This is something that I don't know. For instance, this is in this desert of Namibia, right? There were some papers that, how do you call it, termita? What's in English?
Termite.
Termite were responsible of these kind of circles. but there are no proofs about that. Just to say, you want to say something else, that's fine. You will find this kind of animals for sure, but are they able to just generate this kind of shapes? It's more difficult to prove. That's more difficult to prove.
Yes. This kind of plant, if you cut, for this kind of plant, no, if you cut
Yes.
the plant on two sides,
Yes.
what's remain? It's able to growing.
Yes.
So you have, for example, two species, two plants, but one species.
Yes.
Okay. So it's related in some way in the fact that we don't have much, that we don't have many genotypes, for example, because you can, it's like I can grow outside of me, no?
Yes, we don't. There are, well, that we don't have that many genotypes. This is very interesting because they have sexual reproduction. That
But within self.
within self, yes, can be within self, yes, or with other ones because they just generate fruits. Fruits travel away, and then they grow in whatever place. In fact, just looking at that, biologists were able to identify one of these plants of kilometers of length. And if this is true, taking into account the speed at which they are growing, they might have something like 100,000 years old, something like that, back to the dawn of humanity.
Yes.
Well, that's what they claim. That then it disappeared on the newspapers. Yes, they found the most ancient living being on Earth. Well,
I was wondering about the diversity index,
yes,
and it reminds me of the alternative versions of entropies, which are
yes.
then for other values than Q
Yeah.
are non-extensive. But what's the reason that for higher Q? it doesn't depend
Because
anymore on-
if the exponent is higher than 1 or smaller than 1, most or less abundant species are weighted in a different way. So if you weight especially the most abundant ones, which they dominate, you don't find differences depending on the size that has been sample size. However, if you put much more weight on species that are less abundant, since the number of species, different genotypes appear, is continually increasing with time, then you have something that, of course, changes a lot depending on the number of species because you are able to detect more and more genotypes, depending on the number of sample units because you detect more and more genotypes.
So it tells you something about the shape of your population.
Yes, somehow. This is something that we want to.
And that's,
Or
if it's for high Q, then probably it's minus Q or something.
if it's very high Q, it's probably meaningless because you have just one dominant genotype.
But for the 2 and 3 case here?
Well, but this is typical of what we observe in a Pareto distribution, that there are few species that dominate the whole population, then a large number of them that are just residual or have very spurious presence in the whole system. Something that we're still trying to characterize properly.
Question? Okay, if not, we thank Tomàs again. And